Good day, this is Professor Will, CBMG PhD. Today's Monday, December 20th, 2021. It is 2:04 and 3 seconds, a.m. Eastern Standard Time. Today's research, Nanoparticle Therapeutic. So let's hop to it here. Nanoparticle Therapeutic enhances cancer immunotherapy. Date December 16th, 2021. Source, Wake Forest Baptist Medical Center. Researchers have discovered that the nanoparticle therapeutic enhances cancer immunotherapy and is a possible new approach in treating malignant pleural effusion MPE. Malignant pleural effusion is the accumulation of fluid between the chest wall and lungs and is accompanied by malignant cells and/or tumors. There are more than 200,000 new cases of malignant pleural effusions in the United States each year, and non-small cell lung cancer account for more than 1/3 of cases. Malignant pleural effusions is indicative of late-stage metastatic cancer and is associated with a poor prognosis with an average survival of only 4 to 9 months. Malignant pleural effusions can also severely impact quality of life as it causes breathlessness, pain, weight loss and related physical activity. And in recent clinical trials involving immune checkpoint inhibitors, ICI, or novel immunotherapies, such as anti-PD-1, has shown some encouraging data in patients with malignant pleural effusions. However, only a small number of malignant pleural effusion patients benefit from immunotherapy, and many experience immunotoxicity. The clinical evidence also suggests that malignant pleural effusion comprises abundant tumor-associated immune cells that prevent the body's immune system from recognizing and eliminating the cancer, and this cold immune environment could be a major contributor to the failure of ICI. And to mitigate the immune cold, malignant pleural effusions, researchers developed a nanoparticle called liposomal cyclic dinucleotide for targeted activation of an immune pathway called STING, which reprograms tumor-associated immune cells to active anti-tumor ones. Malignant pleural effusion is often associated with two distinct compartments within the tumor microenvironment, the effusion, and also pleural tumors, which coexist within the pleural cavity. And these two distinct compartments make therapeutic interventions and drug delivery challenging. Upon intrapleural injection in a mouse model, the cold immune environment lessened in not only the effusion space, but also within the tumors. Researchers combined LMP-CDN with an anti-PD-L1 immunotherapy, which drastically reduced the volume of malignant pleural effusion and inhibited tumor growth in both the pleural cavity and lung tissue in mice with malignant pleural effusion, resulting in prolonged survival. Researchers also tested the nanoparticle therapeutic on human malignant pleural effusion tissue samples, and similar effects were observed. Enhanced tumor cell killing by cytotoxic immune cells. And this study demonstrates a possible treatment for malignant pleural effusion. Given the current prognosis of malignant pleural effusion patients, new interventions are needed to not only prolong survival, but also to improve the quality of life. Good day, this is Professor Will, CBMG PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.